Well, hi there, and welcome back. We are going to do a little bit of a review of what happens in the neuron at the synapse and looking at the postsynaptic activity that we were talked about right before we left for break. So here we go. Let's take it away. So synaptic transmission is, as you know, going to require a few things to happen. One is we need an action potential. It's going to travel down the axon and it's going to reach the axon terminal. We're going to have voltage-gated calcium channels, which open, and calcium then is going to enter. So here's the calcium ions, and that is going to facilitate the movement of these vesicles to the postsynaptic membrane and the fusion of those vesicles with the axon terminals membrane so that neurotransmitter can then be released into the cleft or the synaptic gap. Once those neurotransmitters are out there, they can cross the cleft. They can bind then to whatever receptors are found on the postsynaptic side of the neighboring neuron. And then there's two possibilities. One, we can get an excitatory postsynaptic potential, or an EPSP, or we can have an inhibitory postsynaptic potential, or IPSP, depending on which of those receptors are activated on that postsynaptic side. So both of these are going to cause different ions to come in, and then whatever results from that, we get a little bit of a depolarization, or in the case of EPSPs, depolarization of that next neuron, or we could get inhibitory activity, so we'd have actually hyperpolarization. But whatever those things are, and usually it even can be both, it can be a combination, they're going to be moved toward the axon hillock, and the axon hillock then is going to judge whether or not to open the next voltage-gated sodium channel to send an action potential forward. Meanwhile, though, back out in the synapse, we have the neurotransmitter that needs to get inactivated by either enzymes that are going to break it down or transporters that are going to bring it back in by reuptake, recycling, and then the activity's done. There are also things called autoreceptors that I haven't talked about yet, but those are things that also on the presynaptic neuron can influence the amount of neurotransmitter that actually comes into the cleft. So here's a good picture of that, and you can see here's the axon of this presynaptic neuron. You can see the myelin there, they've shown that, and then we have the vesicles that are here already in the terminal, and with their neurotransmitter inside. And there's calcium, so there's our voltage-gated calcium channel. It's going to open, calcium comes in, and activates these to move toward the membrane here at the end of the terminal. They fuse, releasing by exocytosis these neurotransmitters. The neurotransmitter comes out into this little gap, this synaptic cleft, moves across, and if it has a receptor on the other side that it can activate, it will do that. Land on this ligand-gated channel, right? These are ligand-gated. We're now on the postsynaptic side. We're going to have certain ions are going to come in, either sodium or perhaps chloride, depending on what neurotransmitter is activating here. And then that slight either depolarization or hyperpolarization, which is going to happen on this postsynaptic neuron in this dendritic area, it's going to move toward the axon hillock. So this basically, it kind of says in words what I just said. We get the exocytosis, and then the neurotransmitters act as ligands. So there are really two kind of classes of ligands. One are the endogenous. So if you look at endogenous, endo means within, and Gen has to do, like, think about Genesis, but it means it's made. So endogenous ligands are ones that are made by the body. We make them. Our body makes them. It makes these neurotransmitters can be endogenous ligands, or hormones can be endogenous ligands. But there's also ones that we could take in from outside, and those would be exogenous, right? So exo from outside. And those can include things like drugs. These can be types of medications or other things, or even toxins that can be entered from exposure from outside the body. Here's an example. So a synapse, for example, that uses this neurotransmitter, acetylcholine, here's its abbreviation, will have a ligand-gated channel or a binding area on the postsynaptic neuron. And once it goes over across that synapse and it binds to the receptor, then it's going to open a channel. So this is interesting because there are possibilities even with a single neurotransmitter. So in some cases, acetylcholine can act as an excitatory neurotransmitter, and it'll open channels for sodium, as you can see here, or even potassium, which is again positively charged ion. So that's the excitatory part, causing an excitatory postsynaptic potential, or an EPSP. Or acetylcholine can also, in some cases, activate other channels, which would be in the inhibitory ones, opening it up for chloride, which would make it, instead of being depolarized, it's going to hyperpolarize. Okay, we're going to see a little bit more on this coming up. So we've mentioned EPSPs. We know what those are. And these postsynaptic potentials are just very brief changes in that resting potential. So think about the graph we looked at where you saw it at -70, and when it starts the line starts to go up a little bit toward threshold, that's excitatory. So we can have those little wavelets of excitation, and that causes a little bit of local depolarization, and it pushes that neuron closer to threshold and maybe even to threshold. And there can be a little bit of a delay. I'm not going to belabor this synaptic delay piece right now. The other possibility is the inhibitory. So there's IPSP, and we have this hyperpolarization. It's moving in the opposite direction from threshold. So again, thinking of that graph, instead of it starting to move upward, we're going to see that line that would be representing resting potential going down. So instead of -70, it could be -75 or -80. So we're going down, we're getting more negative. And that can result from negatively charged ions coming in, such as chloride. Now here's really an example of what we often see. Remember that neurons they're in communication with hundreds, if not thousands, of other neurons. And so any one of them can have multiple synaptic areas that we can look at. So if this is our main neuron here in the middle, you can see here this neuron is causing an excitatory postsynaptic potential. That's the plus sign right there. So on the graph, here it is, where in this case they're showing -60 as the resting potential. In this case, the postsynaptic neuron is getting this little movement upward, this little wavelet of excitation. That's the EPSP. At the same time, though, at least on this graph, as far as we can tell, at the same time, we have another neuron, though, that is also talking to this neuron. In this case, though, it's releasing an inhibitory neurotransmitter, and so that's the negative sign. And you can see what it's doing is probably opening a chloride channel. And on the graph, if you can see, I'm sorry, my picture is in the way, we get the postsynaptic potential, this IPSP. It's going down, it's going lower than what the resting potential would be. So both of these can happen simultaneously, and as a matter of fact, you can get many, many more inputs to any neuron. And so the job of the axon hillock then is to add up or do summation, to add up the pluses and the minuses, the EPSPs and the IPSPs, and to determine have we reached that threshold in order that would be required to create an action potential, to open that first voltage gate here in this original neuron. So that's this idea of integration. We have to integrate. That's what the role of the axon hillock is to integrate the information that's coming in from all these different neurons and then determine whether or not threshold is reached. So here's another picture that shows a little bit more, and you can see what's happening here. These are different synapses on this particular neuron here. So we have we have the red ones and the blue ones representing excitatory and inhibitory. And these little either wavelets of excitation or of inhibition, the opposite direction, are moving here to the axon hillock. And here it is, it's showing it like a gauge. So in this case, when it does summation, you can see it's on the red side. So this would promote an action potential. So the excitatory activity overrules the inhibitory one. But you can also go the other way. So sometimes neurons will be inhibited because there'll be more inputs that'll be inhibitory. And so that's why this little gauge is showing on the blue. That integration of all of these things is what the axon hillock does. And interestingly, it can happen at various different locations spatially on that neuron, or it can actually also operate in different time spans, which is temporal. So summation can occur in two different ways. One is spatial, which is basically summing up all of the potentials coming in from different parts of the cell. So some of these neurons can actually have synapses in places other than dendrites. And that's one way. So the axon hillock will then do spatial summation, adding everything that's coming in at that particular moment from any part of the cell. And here you can see an example of that. So this is a different type of neuron that we're looking at here, but all of these inputs in here, these all being excitatory, are all coming in to different locations, and that is then being added up here. And in this case, we reach threshold and have an action and generate an action potential. Temporal summation, though, means that sometimes these inputs can happen rapidly, but one right after another at different points in time. So here you can see inputs coming in from the same neuron, but a little bit of a delay between each of those inputs. And that also could cause an action potential. And that's the story. So there's a little bit of a review for you. And now we're going to go into some details on which of the neurotransmitters are involved. This is what you're doing your research on. Which of these neurotransmitters are involved with excitatory or inhibitory activity, and not just anywhere, but in particular areas in the brain, and what that results in. So there you go.